Okay, so I will start by reading this question here. So, erythritol, a natural sugar abundant in fruits and fermenting foods, is about 65% as sweet as table sugar. So, erythritol, 65% as sweet. I don't know if these numbers are important. I will write them down though. Ooh, 95% fewer calories. Nice. Tooth-friendly and generally devoid of negative side effects as a human consumable product. So, pathogenic Brucella, I'm guessing it's some type of bacterium. Pathogenic, so I'm guessing that it must be pathogenic on maybe pathogenic on the plants that make the fruit, pathogenic on fruit, maybe. So, pathogenic Brucella strains that catabolize erythritol contain 4 closely spaced genes. So, Brucella catabolize or break down erythritol. So it must use this as a carbon source, as a sugar. So it eats erythritol. And it's probably going to be like the lac-operon here. So let's take a look. 4 closely spaced genes all involved in erythritol metabolism. One of the 4 genes, ERYD, so 4 genes, there's 1, 2, I'm going to assume ERD is first, maybe not, encodes a product that represses the expression of the other 3 genes. Okay. So if this is anything like the lac-operon, then ERD is going to be expressed from a different promoter. So for now, I'm going to assume ERD is over here, and we have a promoter and operator over here. Okay. So erythritol catabolism is stimulated by erythritol. Okay, so let's give erythritol the same kind of structure as we give to lactose. So you have this in the cell, and then so expression of the genes is stimulated. Present a simple regulatory model to account for the regulation of erythritol catabolism in Brucella. Does the system appear to be under inducible or repressible control? Okay, so a simple model is we have our 4 genes here. Let's call this gene A, gene B, gene C. This was ear D, so this can be ear A, ear B, ear C. Can be an operator here with a binding site for the repressor. So what did we say for ear D? Again, encodes a product that represses the expression of the other 3 genes. Okay, so let's make it look just like lac-I. So we know that, I think a key line in this question is that catabolism is stimulated by erythritol. So here's erythritol. We know that we add this, if we add this to the cell, catabolism is stimulated. That means when this is here on the repressor, then the repressor is no longer going to bind the operator, and we're going to transcribe these genes here. Okay, we're calling this ear D, repressor. Normally it is repressing. We add erythritol to the cell. Allosteric shift, can't bind anymore. As a result, these can be transcribed, and erythritol catabolism is stimulated. Okay, so I think that one is pretty simple. It's essentially the same thing as the regulation of the lac operon by the lac-I repressor in the presence of lactose. So no tricks there as far as I can tell. And as a result, it appears to be under inducible control, right? So it is inducible. We add erythritol. Erythritol binds to the repressor, and the repressor does not repress anymore. So the operon is induced by the presence of erythritol.